Hi, and welcome to the Match Game. This series will guide you through various strategies to identify your unknown family finder matches. I'm your host, Katy Rowe-Schurwanz, and I know y'all are excited to play, so let's get started. Welcome to part 2. In part 1, we reviewed the clues in your match list that are more closely tied to traditional genealogy, shared family trees, and shared ancestral surnames. This part digs into the clues that are found in your DNA and how to use various tools and features to easily tie those to your genealogy, form match groups, and prove the connections you hypothesized from your paper trail, and discover new branches on your family tree. So let's get started. You don't need a biology or genetics degree to understand and use these tools and apply them to your genealogy research, even though it might seem like that at first glance. In the next few slides, I'll walk you through how to take the science and turn it into your family stories. We'll review how to find and form match groups out of matches who not just share DNA, but overlapping DNA segments, meaning that those segments were passed down from the same shared ancestor to your whole match group. We'll also look at how DNA passed down in different patterns can help, like xDNA, yDNA, and mitochondrial DNA, often abbreviated as mtDNA. And I'll review a few strategies that I use to organize my research and check my work. Before we dig into how, let's go over why we're going to be following these next steps. By triangulating matches, which is forming groups of at least 3 matches who all match each other and all overlap on the same DNA segment with each other, you can deduce that the members of that match group all descend from the same common ancestor who passed down that segment of DNA to each of them and to you. Through the tools we covered in part 1, you can determine who that ancestor was. Triangulation lets you identify which ancestor gave you which DNA segments and who on your match list also inherited that same segment from that same ancestor, allowing you to confirm how you're related to them. Your shared matches are what is going to unlock everything for you. These are matches that share DNA with both you and another specific match. So if you match Aunt Sue on your mom's side, you and Aunt Sue will have other matches that share DNA with both of you, implying that those other matches are also going to be on your mom's side. There's a little bit more to it, but that's the general idea. People who share the same DNA with each other share the same ancestor with each other. Grab a match from your list that you want to investigate. If there are known matches that you've linked on your tree, or you at least know who your common ancestor with them is, start with one of those. If you don't have any of those, or once you've run out of them, then start with the closest unknown match, and then the next closest, and so on. Check the box to the left of that match's name. This is going to save them for comparison in either the Matrix or the Chromosome Browser, and that's a couple of steps from now, so you're just saving them right now. We'll get to what to do in the Matrix and the Chromosome Browser in a few slides. On the right side of the match is an icon that looks like the outline of the top half of 2 people. That's the in common with and not in common with searches. In common with will give you a list of matches that match both you and the selected match. From here, you're going to select in common with matches to compare in the Matrix and the Chromosome Browser to create groups that not just match each other, but match each other on the same DNA segments. You can select up to 10 for the Matrix, but only up to 7 for the Chromosome Browser. So we're going to select 6 more matches to compare. So which are the best matches to choose? And this is where you're going to start combining the skills from part 1 with these new skills. If this is a match that you have linked on your tree, they'll be assigned to a parental side, paternal or maternal, with the family matching tool. This helps you filter out which matches you don't want to compare. If the match you started with is on your paternal side, then you'll want to ignore any matches that are assigned to your maternal side. Stick with either paternal assigned matches or unassigned matches. You may see matches that have been assigned to the other side, but those are going to be matches that share one ancestor with you and a different ancestor with your selected match. They aren't going to fit in your match group that you're forming right now. If you're starting with a known match, so someone that you've identified your shared ancestor with, or someone you have a hypothesis about how you're connected, look for shared matches that share ancestral surnames on that same ancestral line. On your match page, after you've run your in common with search, you can still use the search field at the top right to search by ancestral surname. You'll want to avoid matches who have immediate family relationships with each other. Sometimes you won't know that they do until you compare them in the matrix, and I'll show you how to do that in a couple of slides. But if you know already, only include one immediate family member. And if all else fails, just go down the list and compare 7 at a time. Select each match by checking the box to the left of their name. Once you've chosen them, click Compare Chromosome Browser in the bar at the bottom of the match page. When you get to the Chromosome Browser, all of your selected matches will be ready to analyze. The Chromosome Browser lets you visualize which segments you share with your matches and identify groups that overlap on the same segments with each other. Each match will have a color and a row for each chromosome. So the first match in the list is assigned blue, and there'll be the top row on the Chromosome Browser. The second match is pink, and there'll be the second row, and so on. With the matches you're comparing, we're looking for ones that have the same section of the same chromosome colored in. That's what overlapping segments will look like. Every highlight is what the match shares with you, but we're looking for areas where multiple matches share that same segment with you. The overlap doesn't need to be perfect. You can share a longer segment with some matches and a shorter segment with others, but what is important is that there is a decent sized section that overlaps. There is one tricky thing here, and we're going to compare in the matrix too because of it. The Chromosome Browser doesn't differentiate between the chromosome you inherited from your mother and the chromosome you inherited from your father. So it's possible that these overlapping segments aren't really overlapping. One match may share those segments with you on the chromosome you inherited from your mother, and another may share those same segments with you on the chromosome you inherited from your father. This is one of the reasons why you're not selecting matches that are assigned to different parental sides than your main match, the one you did the in common with search on to begin with. If all of your matches you're comparing are assigned to the same parental side, this is less likely to be the case, but you still need to double check. So let's go over to the matrix and do that. The matrix lets you compare groups of matches and see if they match each other, how they match each other, and how many DNA segments they share with each other. In common with will tell you if a match matches you and the one you started with, but the matrix will tell you how you, that match, and each of your shared matches match each other. You'll want to group together matches that match not just you and the first selected match, but also match each other as well. Those are going to be the most likely to share overlapping DNA segments with the same common ancestor or on the same ancestral line. On the previous screen, 3 out of the 7 shared matches we started with overlapped on the same segment on chromosome 9. The matrix is going to help us find out more about how those 3 matches match each other. In the top half of the page, add those 3 matches to the list for comparison and select which data to display for that comparison. There are 3 options for the comparison. Relationship range will help you do a couple of things. It can weed out immediate family members from your match group, and in combination with the total centimorgan shared option, it can help you identify how you all fit on the family tree and descend from a common ancestor. There's a manual tool at DNA Painter where you can test out various tree configurations. It's called What Are the Odds, or WATO. And then there's an auto tool at Genetic Affairs, which I'll show you in a couple of slides that can help you with that as well. The number of shared segments is going to help you determine if the segments your match group overlaps with you on, they also overlap with each other on. If you and your comparison group all share one overlapping segment with each other, then it's the same segment. That segment was passed down from the same ancestor to you and to each of them. That's what you're trying to determine here. That's triangulation. Now you just have to figure out which ancestor it was that gave y'all all that DNA segment. If they share more than one segment with each other, though, you don't know if one or none of those are the same segment that you share with them. The best way to be certain is to reach out to the matches and ask them if they share that segment with each other. In this example we've been using, I did reach out to the matches that share two segments with each other, and one of those segments is the one that they share with me, and so I was able to confirm which ancestor we all share by looking at their shared family trees, and then I was able to link all three of them on my family tree. Clustering is another tool that can help you more quickly form match groups and analyze them. But there's a couple different types of clustering, and which type you're using will determine how much work it's doing for you. So there are two types of clustering. The first is shared match clustering. Shared match clustering is like doing an in common with search for a bunch of your matches at once. You'll end up with a chart like you see here with different groups of matches that are all in common with each other. Each of those little colored squares is a match. This is what you'll find at Genetic Affairs, which is shown on this slide. Also MyHeritage, GEDmatch, and Ancestry. You can also do this manually with the LEADS method, and you can find information about how to use the LEADS method in the syllabus for this class. One thing that I will add here is that clustering tools use a lot of data and require a lot of processing, so they're fairly expensive to create and run. Because of this, most third parties and testing companies put these types of tools behind a subscription, including Genetic Affairs, which I'm using here. The second type of clustering is triangulation clustering. Triangulation clustering takes things a step further. Not only is it creating groups of matches that are in common with each other, but those groups also share the same overlapping segments with each other. So this is what you're doing manually with the Chromosome Browser and the Matrix. Genetic Affairs has an auto segment tool that will do this, which is shown here. The results will also include which parental side each match is assigned to. So you'll get a file that's got the chart with all the little colored squares like we saw in the previous slide, but you'll also get a list of which matches are in each cluster, how many segments they share, and what the size of those segments are. While this is telling me that these matches match each other and they overlap on the same segments, I still want to look at them in the Chromosome Browser and the Matrix. One, because it double checks it, but also it gives me data that I'll need to keep track of them and which ancestor passed down that segment, and I'll show you how to do that in a bit on DNA Painter. Genetic Affairs also has a couple of other tools that are pretty helpful, and I'll talk about these tools again in part 3. Their auto tree tool will look at each cluster and find common ancestors and locations in their shared family trees. And the auto kinship tool will build a predicted family tree for your clusters based on how much DNA you share with each clustered match and how they match each other, how much they share with each other. This is the automated tool that I hinted at back when we were talking about the matrix and relationship ranges a couple of slides ago. These types of tools can help you quickly identify a common ancestor or a generation in your tree to examine to find the common ancestor. You can then take that cluster, compare them in the Chromosome Browser, and identify which segments belong to that ancestor if they overlap, and the paper trail checks out, of course. In the next few slides, we're going to talk about matching on DNA with different inheritance patterns than autosomal DNA, and how that can help you identify which ancestor you share with a match. We'll start with xDNA and your X matches. The Family Finder also tests your X chromosome, and on your match list, you'll be able to see if someone is an X match. This will mean that you share at least one segment of 10 centimorgans or more on the X chromosome with them. Centimorgans, which are typically abbreviated with a lowercase c and uppercase m, are the unit of measurement for how much autosomal DNA you share. There are fewer ancestors you would have inherited xDNA from, so this can be another way to narrow your list of potential most recent common ancestors with your matches down. The fan charts on this slide highlight which ancestors those can be. So men have one X chromosome and women have two X chromosomes. Fathers will pass their X chromosome down to their daughters. There's not another X for it to recombine with, so it gets passed down without any changes. They do not pass an X chromosome down to their sons. Mothers, however, will pass one X chromosome down to all of their children, both sons and daughters. Because women have two X chromosomes, it doesn't get passed down without changes like a father's does. Instead, it remixes itself before getting passed down like chromosomes 1 through 22 do. You're going to use the same skills you did for regular matches to analyze your X matches. The list of potential ancestors you could share is going to be smaller. And because there's less DNA to compare on the X chromosome than on the other chromosomes, you're going to want to look at larger segments than you were using previously. FamilyTreeDNA has a higher threshold for the amount of shared DNA required to be an X match versus to be a regular match for that reason. So you'll typically want to double the segment size. So if for chromosomes 1 through 22, you're looking at segments of 20 centimorgans or above for normal matches, you'll want to do 40 centimorgans or above for X chromosome segments. Direct line testing can also be a way to double or triple check the ancestral line you assigned your match group to. If you have multiple people in your match group that are direct paternal or direct maternal descendants of the ancestral pair you'll share, so all fathers from them to the ancestor or all mothers from them to the ancestor, then they should also match on Y-DNA for paternal or mtDNA for maternal. Y-DNA and mtDNA differ from autosomal DNA in how they're passed down. Y-DNA goes from father to son, and mtDNA goes from mother to child. Not only are these only coming from one ancestral line each, they get passed down with no or very few changes over generations. So there's no random recombination. So your Y-DNA and your fifth great-grandfather's Y-DNA may be exactly the same, or your mtDNA and your eighth great-grandmother's mtDNA may be exactly the same, or there might be a couple of differences. So 2 or more matches who are both direct patrilineal descendants on paper should also be Y-DNA matches and share the same Y-DNA haplogroup. So they'll be placed on the same branch on the genetic family tree of all humans. And 2 or more matches that are both direct matrilineal descendants on paper should be mtDNA matches and share the same mtDNA haplogroup. So they'll be placed on the same branch on the genetic family tree of all humans. Because of this, Y-DNA and mtDNA testing can provide confirmation that your match group can be assigned to a specific ancestor. And it doesn't even need to go too deep in Y-DNA or mtDNA analysis to do that. You just need to know, do you match, yes or no? And while no usually isn't the desired answer, it is an answer. They're not related that way, so go back to the drawing board. It's going to be a different ancestor. As you're confirming who your most recent common ancestor with your previously unknown matches is, you'll want to continue building out your family tree to include them and then link them on your tree for family matching like we covered in part 1. This will assign more matches to your paternal and or maternal sides, helping you to continue to identify more matches. If a new match is assigned to this side, it's going to share the same common ancestral line as one of the matches that you have previously linked. So if you figure out an unknown match that's also descended from great-grandmother Beatrice on your maternal side, and you link them on your family tree, all the new matches that get assigned to your maternal side from that one link will also be descended from great-grandma Beatrice or one of her ancestors. The more matches you link, the more ancestral lines you'll have covered, and the more quickly you'll be able to identify the next unknown match, and the next, and the next, and the next. On your match list, to the right of the In Common With icon and the pedigree icon, next to each match is a notes icon. These are for you to leave notes about your connection to your matches. So any hypotheses you have about your connection, where you're at in your research, and so on. I'll also leave a note here about what I find in their tree. Is it private? Does it only have a couple of generations? Did they add one to another site that may have more information? Things that can help me when I come back and I'm looking at this match again. I also use a spreadsheet to keep track of which matches I've linked to which ancestors. I have a separate tab for each family member that I've tested. In my tabs, I create a row for each ancestral pair. The next column is their locations, and then I have a column for each match that I've linked to that ancestral pair, and one for matches that might be linked to that ancestral pair, but I haven't confirmed it yet. As you're confirming which ancestors your unknown matches share with you, you can also use the chromosome mapping at DNA Painter to keep track of which segments belong to which ancestors. To get the segment data, you'll need to do this. You'll go to the Chromosome Browser on FamilyTreeDNA. You'll add your match group there and compare them. Then you'll want to switch from the chromosome view that we looked at earlier to the detailed segment view, which is the second tab in the Chromosome Browser. Highlight the row or rows for one match. Right-click with your mouse and select Copy. You're going to paste that data on DNA Painter and then come back and do the next match and the next match and the next. On DNA Painter, you'll want to go to the chromosome mapping tool. When you've created a map, click Paint a New Match near the top right. In the box that opens up, right-click with your mouse and select Paste, and then click Save Match Now. In the next step, you'll select if you know how you're related to the match or not. You'll enter the match's name. If you know the ancestor and you've already added them to your map already, you can select them. If this is the first match that you're adding that's related to that ancestor, then you can add the new ancestor and select a color for them. When you're ready, you'll click Save Match, and then you'll go back, copy the segment data for your next match, and do this all over again. You'll end up with something like the image number 3 here for a match group that you've triangulated and painted for a specific ancestor. So you can see which segments on which chromosomes that ancestor passed down to you and your match group. And eventually you'll end up with a visual representation of which segments you inherited from all identified ancestors. When I'm comparing new unidentified matches that get added to my list, and I'm looking at them in the Chromosome Browser and the Matrix, I keep my chromosome map at DNA Painter open in another tab. Because of the other match groups that I've added, I can form a hypothesis about which ancestral lines my new matches share with me based on where they overlap with the ancestors that I've previously assigned those same segments to. And using the other tools that we've covered in these first 2 parts of the series, I can determine which of those lines it is and which ancestor on that line, and I can add them here and add them to my spreadsheet and link them on my family tree. We've covered a lot of topics here, and luckily it's virtual, so you can come back and watch it again and again and again and again. But there's also some other resources out there on the internet to help you with the things that I've covered here. I've listed a few of my favorites here, and you can also find them in the syllabus. FamilyTreeDNA's Help Center and blog have a ton of great information. Roberta Estes' book, The Complete Guide to FamilyTreeDNA, truly is a complete guide, and her blog, DNA Explained, is a treasure trove of genetic genealogy tips and tricks. And I love Diane Southard's Your DNA Guide. She's got a blog, books, quick guides, and offers great webinars and courses. You can also find more content on some of the tools in our virtual booth here at RootStack. There's one on the Matrix and the Chromosome Browser, and one on xDNA matching. And you can find tutorials for tools from Genetic Affairs and DNA Painter on their respective websites. Join me for part 3, where we tackle the scenarios that aren't perfect. We'll be looking at multiple relationships with your matches, endogamy, and unknown parentage.